My dear students, welcome to my today's online class on applications of PCR technique. In my previous class, I have taught you the PCR technology, how exactly it is done. And now we have to learn what are the applications of PCR in molecular biology and biotechnology. Okay. I am Dr. Meera BK, Associate Professor and Head, Department of Zoology, Maharani Science College for Women, Palace Road, Bengaluru 1. There is mobile number and my mail ID being depicted on this slide. After the talk, after the class, any queries, don't hesitate to contact me. And this is an automated machine of PCR. Now we shall study the applications of PCR. Some of the applications of PCR can be briefly discussed like this. First one is PCR in clinical diagnosis. That means applications in medicine. See, the PCR, as I have taught you, is highly specific and sensitive technique. Therefore, is useful for the diagnosis of various diseases in humans. This includes inherited diseases or genetic disorders, viral infections, bacterial infections, etc. Let us study one by one. See, there is a technique called RFLP, restriction fragment length polymorphism, which I will be teaching you in next coming classes. The three techniques, RFLP, RFPD and one more. Okay. Now, normally the occurrence of genetic disease is frequently identified by this technique, RFLP. Okay. But RFLP can be employed only when there is a mutation resulting in a detectable change in the length of restriction fragment. So, if that mutation doesn't result in the noticeable change in the length of restriction fragment, then RFLP cannot be employed for diagnosis of genetic diseases. Okay. And for your information, many genetic diseases occur without the involvement of RFLP. You may not see, right? So, if that is the case, what is the solution when RFLP cannot be employed? For such disorders, PCR can be employed as it provides direct information of the DNA. Okay. This is done by amplification of the DNA of the relevant region, which is followed by the direct analysis of the PCR products. Thus, the genetic diseases, in those cases where RFLP cannot be employed, you can readily go for PCR technique. Okay. Then, secondly, PCR is useful for prenatal diagnosis of inherited diseases. Okay. PCR is useful for prenatal diagnosis of inherited diseases, where it is employed by using chorionic villus samples. You will take the cell samples from chorionic villi, right, that is part of placenta, or cells from amniocentesis. Okay. Diseases like heritable diseases like sickle cell anemia, beta thalassemia, phenylketonuria, etc. can be diagnosed prenatally by using PCR technique. So, prenatal diagnosis of inherited diseases is possible by PCR technique. Then, diagnosis of retroviral infections. See, here is a case where you can do PCR from cDNA, that is complementary DNA, which forms a valuable tool for diagnosis and monitoring of retroviral infections. Please note, these retroviruses will have RNA as the genomic material. Therefore, from RNA, you have to synthesize cDNA, that is complementary DNA, and then from cDNA you can have a double-stranded DNA. Okay. So, PCR from this cDNA forms a valuable tool for monitoring retroviral infections like HIV infection, that is AIDS, and then right now we are facing a pathetic situation of COVID-19 pandemic, right? That also belongs to this group, right? For which we make use of one particular type of PCR, that is RT-PCR, reverse transcription PCR is used. Okay. So, you can diagnose retroviral infections by PCR technique. Apart from viral infections, bacterial infections are also diagnosed. Best example is TB, tuberculosis, caused by bacterium, Mycobacterium tuberculosis. Okay. Apart from retroviral infections, bacterial infections, PCR is helpful in diagnosis of cancers, especially virally induced cancers like cervical cancer, that is cancer of cervix of uterus. Okay. Uterus has a small part called cervical region. So, cancer can happen to that region by the infection of a virus called HPV, human papilloma virus, and that can be detected by PCR. So, if cancer is diagnosed in earlier stages, treatment becomes easy and survival rate also will increase. Okay. Then, some cancers, which occur due to chromosomal translocation, especially chromosome number 14 and 18 in follicular lymphoma. There is a cancer called follicular lymphoma. Lymphoma is the cancer of lymph glands. Okay. So, here cancers occur due to chromosomal translocation. By now you must have studied addition, deletion, translocation, etc. in your genetics class. So, here cancer is due to the chromosomal translocation of number 14 and 18 causing follicular lymphoma, and genes involving in causing such cancers can be diagnosed by PCR. Okay. Then PCR is also employed in sex determination of embryos, sex of humans and also livestock. Okay. Livestock fertilized in vitro, that means you have carried out fertilization in the laboratory, can be identified or determined by PCR using primers and DNA probes which are specific for sex chromosomes. PCR is also useful to detect sex-linked disorders in fertilized embryos. So, PCR can be employed in sex determination of embryos both in humans and livestock. There is a need to do so even in livestock, okay, other than humans, and PCR is also useful to detect sex-linked disorders in fertilized embryos. Okay. So, these are clinical applications of PCR. Then, PCR in DNA sequencing. It is a simpler and quicker technique available to date to amplify the DNA. Therefore, it is conveniently used for sequencing, DNA sequencing, and if you are aiming at DNA sequencing using PCR technique, you have to use single strands of DNA. If you have double-stranded DNA, you have to renature it and then you have to separate out the templates. Then you can use the single strands of DNA for sequence analysis. Then, fourth application is PCR in gene manipulation and expression studies. Please note one thing, that the advantage of PCR is that the primers what you use in the second step of PCR, that is annealing or renaturation, need not have to have complementary sequences for the target DNA. They need not have to have complementary sequences for target DNA. Therefore, the sequence of nucleotides in a piece of target DNA can be manipulated and amplified by PCR. This particular technique is known as site-specific mutagenesis. It is so nicely done in the sense a single nucleotide can be replaced. Say for example, adenine can be replaced by thymidine. You can create mutation artificially. Okay, by a single specific mutation can be done by this method. The coding sequence can be altered to synthesize the proteins of interest. With this background, I want to tell you that gene manipulations are important in understanding the effects of promoters and inhibitors in gene expression. I want you to go back to your previous semesters where you have learnt gene expression in prokaryotes and gene expression in eukaryotes. There you must have studied the concept of promoters and inhibitors, right? So gene manipulations are important in understanding the effect of promoters and inhibitors in gene expression, and that you can do by PCR. Okay, so thus PCR is helpful in gene manipulation and expression studies. Then PCR is important in the study of mRNAs. You know that the DNA is decoded, right, when expressing to mRNA, right? So you can study the mRNAs which are the products of gene expression, and this is carried out by a particular type of PCR called RT-PCR, reverse transcription PCR. Okay, then PCR in comparative studies of genomes. The differences in the genomic component of two organisms can be measured by PCR with random primers. Okay, two genomes from closely related organisms are expected to yield more similar bands. This is done by a technique called RAPD, random amplified polymorphic DNA, which I am going to teach you in next coming classes. So by employing RAPD, the two genomes from closely related organisms can be studied, and if they are very closely related, they are expected to yield more similar bands. Then PCR is a very important tool in the study of evolutionary biology nowadays, more specifically referred to as phylogenetics. Okay, so PCR has its application even in evolutionary biology at molecular level, which we call it as phylogenetics. Then PCR can amplify even minute quantities of DNA from any source like hair, mummified tissues, bones, or any fossilized material. Thus, PCR in fact has revolutionized the studies in paleontology and archaeology. Okay, so there lies the significance of PCR in the study of paleontology and archaeology. Then PCR in forensic medicine. A single DNA molecule from any source like blood stains, hair,